Okay, we're going to have a bit of a look at our gene technology toolbox. So we're going to have a look at the tools that scientists might use when they're manipulating DNA and they're working in biotechnology or genetic engineering. We're going to specifically look at the molecular tools that they use rather than the equipment. The things like the PCR machine or the gel electrophoresis equipment are things that we can look at in a bit of detail when we look at the techniques that these tools are used for. So first off, let's have a look and have a look what the scientists are working with. They're working with DNA, so sections of DNA from the organism or the bacteria that they're studying. But they more specifically in bacteria quite often work with these plasmids, these small rings of DNA that they can be easily passed from one bacteria to another. They reproduce quite quickly in the bacteria, and they're relatively short, and so we can know the sequence of them quite easily. There's also the chromosomal bacteria in bacteria as well. Now, if scientists are building DNA, they're going to need plenty of nucleotides. We know that the different nucleotides have different nitrogenous bases, but they're going to need a solution full of these nucleotides if they're going to be building molecules of DNA. So let's have a look at the basic molecular tools. Now the first one here is restriction enzymes, and they have the job of cutting DNA, and they can cut DNA in specific spots. So the scientists can control where in the DNA it's cut and what that cut looks like. There's also DNA ligase, which is the enzyme that's used to join DNA sections back together, and we know a little bit about that from a study of DNA replication, where we looked at how ligase was used to join the Okazaki fragments. Another one we know from DNA replication is DNA polymerase, and that's used to add nucleotides. And if we're going to be building strands of DNA, we're going to need some of that in the solution as well. There's a specific one that we'll look at a bit later called Taq polymerase. And finally, here we have primers, and the primers are small sections of DNA or an RNA that attach onto the DNA to start the building of the DNA. The nucleotides just won't come along and start at any spot by themselves. They need these little start sequences to make things happen. So first off, let's have a look at restriction enzymes in a bit more detail. Now, the example I have here is one called Echo R1, and it cuts at a particular sequence. Whenever it sees AATT, it does a little cut like this, 4 nucleotides long, and results in a little step which we call a sticky end. And this little overhang is what it creates. Now, it might do this cut along AATT several times. It'll do it every time it sees that particular sequence. And it'll create these sticky ends, and these sticky ends are really useful. If we know that that's what the end is going to be like, we can control what this piece of cut DNA can join onto. So here we have the same sort of thing. The sticky ends produced by Echo R1. When it cuts, we say that it digests the DNA when it cuts it. We have another example of a different restriction enzyme, and this restriction enzyme is creating a blunt end. And this end doesn't have an overhang. Might be fine for cutting up the DNA, but as far as having control about joining it back together again, these sorts of sticky ends are a lot better, a lot more useful. So here are some examples of some restriction enzymes. And we can see that some of them are producing sticky ends, some of them are producing blunt ends. The sticky ends can be different sequences, and they can have a different number of nucleotides in the overhang. They all seem to have quite funny names, but quite often it's from the source organism. So this one here, Echo R1, is from an E. coli bacteria, getting the eco in its name, Echo. And R1, well, I imagine that if they found another enzyme from this E. coli, and that it was a particular restriction enzyme they wanted to use, they might call it Echo R2. Now, scientists having all this variety of restriction enzymes that can all cut in specific spots and specific places is extremely useful for them. It gives them a lot of control about cutting the sequences and cutting particular genes out or particular length of sequences, but also a lot of control about joining them back together. So an example here might be if we want to insert a foreign bit of DNA in a bacterial plasmid. We could use Echo R1, for example, to cut the DNA, and it would cut every time it's CAACT, and it would cut into particular segments, and we could get one of these segments and insert it into the plasmid. If we cut the plasmid with Echo R1 as well, the overhang is going to be the same, and with the same sort of overhang or sticky end on the plasmid as on the bit of DNA that we want to insert, we can actually join them together, and we call that annealing. Now, I mentioned before that we already know that a polymerase adds nucleotides. In the lab, a really useful tool for us is one called Taq polymerase, and it comes from a bacteria that lives in thermal springs, and this bacteria is able to survive at really high temperatures, which means its enzymes can work at really high temperatures. So we use its polymerase in the lab because a lot of the samples of DNA, we have some processes that we do where we actually need to heat the DNA samples up together, and if we can have a polymerase in there that can handle those high temperatures and isn't denatured, that's really beneficial. I'll mention two more that are a bit more specific. One is gene probes, and gene probes are small sequences of DNA or RNA that are complementary, and when we mix them with DNA, we'll know where they attach and where they attach to the strands. If we mark them with radioactive marker or a fluorescent marker, we can see if that probe has attached. And a great way to detect if a particular gene or sequence is present. We might be able to check for the gene for a particular disease, and if the probe attaches, we know that the gene that creates that disease is present. The very last one we want to look at is something called reverse transcriptase, and it does exactly as the name suggests. It runs transcription in reverse. So normally transcription runs from making from DNA making RNA, and this can do the reverse. It can make DNA from RNA. So if we knew a particular amino acid sequence in a protein, or we had the codons in the messenger RNA, we could then use this enzyme to create a copy of the DNA that could be inserted into an organism or a plasmid. And when that's inserted there, we know that when it starts to function, it'll produce this particular messenger RNA and then this resulting protein. Okay, so it allows us to do sort of like a reverse design sort of process. Okay, we can insert the DNA that we know what it'll make. Okay, good luck with studying gene technology. We've just introduced a lot of the tools. The next stage of things is to look at what techniques these are used for, and we'll look at things like PCR and gel electrophoresis. And the final stage is to look at how we might apply this knowledge in the real world. What sort of uses do we actually have for all this sort of stuff? Okay, so enjoy.